Hello friends, in last class we have discussed about DNA and its different aspects like transcription and translation. In the present class we are going to discuss about RNA, its different types and genetic code. It's one of the sub theme, simply types of RNAs. Of course, RNA simply stands for it is a polymer of ribonucleotides. It's a polymer of ribonucleotides. The essential distinction between RNA and DNA is its sugar component. That means pentose. The kind of sugar in RNA is D-ribose. That means C5H10O5. Whereas in case of DNA, it is 2-deoxyribose C5H10O4. We have already discussed. Say the D-ribose in RNA is C5H10O5. Is D-ribose, whereas in DNA is a small difference. At second carbon there will be no hydroxyl group. That's why due to deoxyribose. That's the main essential distinction between DNA and the RNA. C2 is 2-deoxyribose. D-ribose, is a point. Apart from this, what we call pentose differentiation, we also have a pyrimidine differentiation. In case of RNA, uracil is present, which is in place of thymine, for DNA. We all discussed on the structures what are different on. It is 2,4-dioxypyrimidine. Simply 2,4-dioxypyrimidine is uracil. This is about different things. The basic distinction between this is general structure, Whereas in case of thymine, it is 2,4-deoxy-5-methylpyrimidine. That's it. So in place of thymine, uracil is present in case of RNA. And of course, the most important point, RNA is a single-stranded component. It's a single-stranded component. There is a possibility of double-stranded nature when there is a proximity of complementary base pairing. Okay, it's about different basics of RNA, and we will elaborate about different types of RNAs. Basing on the occupancy or the percentage, there are majorly three broad categories of RNA. Three broad categories. Number one, messenger RNA. Number two, transfer RNA. Number three, ribosomal RNA. With respect to cellular occupancy or percentage, it is just 5 to 10%, it is 10 to 20%, it is 50 to 80%. Apart from these regular or conventional major types of RNA, we also have other types of RNA like snRNA, small nuclear RNA, snRNA, small nucleolar, it is nuclear, it is nucleolar RNA, hnRNA, that is heterogeneous nuclear RNA. What we call primary transcript. In case of eukaryotes, the RNA synthesized in transcription is not mature. It has to subjected to certain post-transcriptional modifications. That means premature RNA. Primary transcript is called heterogeneous nuclear RNA. It's about minor issues. Say snRNA involved in mRNA processing, small nucleolar RNA involved in rRNA processing, hnRNA is primary transcript. But majorly these are different types: mRNA, tRNA and rRNA. Okay, we'll discuss point by point. First, mRNA. That name it implied, messenger RNA. Say it's a DNA strand, it's a template strand. This is mRNA. What we call transcription. The role of mRNA, the message, it will carry the message, or that means by copying the genetic information present in DNA in the way of an intermediate complementary mRNA, and which will aids in translating the DNA language into the protein language, what we call simply translation, through ribosomal machinery. This is a major functional head of mRNA. Whereas the most important point is, in case of eukaryotes, there are certain post-transcriptional modifications which we have to discuss about. What we call post-transcriptional modifications. Majorly 5' methyl cap. This is 5' and methyl cap means addition of 7-methyl guanosine. 7-methyl guanosine through an enzyme called guanosyl transferase. Guanosyl transferase. The kind of linkage is 5' to 5' covalent, as 5-adenosyl methionine is the donor. It prevents from the attack of 5' exonucleases. This is very important point. The 5' capping, just like we wear helmet during driving, the same mRNA has to get protected from the hydrolytic activity of 5' exonucleases. For that, methylation is essential. And how it is done? There is an enzyme called guanosyl transferase. It will add the guanosine residue to 5' covalent bond in the 5' end, and later onwards methylation is done, what we call 7-methyl guanosine, and the donor is simply 5-adenosyl methionine. That's how the first important post-transcriptional modification in case of eukaryotes is done. Then the second, in this case only in eukaryotes, okay? In case of prokaryotes there is no need of such things, because what we call coupled transcription and translation. When the transcription is, that means translation starts before the end of transcription, because there is no need of such things. The second one is called polyadenine tail, simply PAA tail, which prevent from 3' exonucleases activity. At the same time, stability of RNA, that means mRNA, and direction from nucleus to cytoplasm is very important. Say magnification, there is a poly-A tail. It will be like back helmet or a rear helmet which protect the mRNA from the 3' exonuclease activity, and will direct the mRNA from nucleus out to the outside cytoplasm for translation machinery. This is very important. Apart from this, we also know there is RNA splicing. RNA splicing is very important because we have already discussed when we are discussing about types of RNAs. The first mRNA which is formed is called primary transcript or heterogeneous nuclear RNA. It contains some non-coding sequences, for example, exons or coding sequences, introns or non-coding sequences. Likewise, exon and introns. These introns has to be removed by spliceosomes. Therefore, exon should get attached. This is called mature RNA. This is called RNA splicing. This is the important part. You have to form a coding sequences of mRNA. This is essential. Then apart from RNA splicing, there is another called RNA editing. RNA editing. It also modifies the nucleotide sequence simply by insertion, substitution, deletion. which resulting in changing or modification or amendment of the encoding framework of mRNA so that a different polypeptide may emerge, that means may synthesize. That means it is manipulating the DNA information. Actually, there is what we call collinearity. There should be a collinearity between the DNA sequence in the gene with the of the polypeptide sequence through an intermediate complementary mRNA. But what happens in the way of RNA editing is the sequence in RNA will get changed. So a different polypeptide may come. It also happens in the way of post-transcription modification in eukaryotes, okay? And it's about different aspects of mRNA. Then the second important component type of mRNA is ribosomal RNA. rRNA simply called. Its major role is structural framework of ribosomes. How? In case of prokaryotes, it's a different kind than that of eukaryotes. The prokaryotic ribosome is called 70S, whereas in eukaryotes it's called 80S, 30S and 50S. And this 30S subunit contains 16S rRNA and 21 polypeptides, whereas the 50S contains 23S rRNA, 5S rRNA plus 34 polypeptides, whereas the 80S contains 60S, 40S and 60S. The 40S contains 18S rRNA and 33 polypeptides, whereas 60S contains 28S rRNA, 5.8S rRNA and 5S rRNA. Then polypeptides are 49 polypeptides nearly, It's about the structural framework. That means here the ribosomal RNA is coupled with the complex of core protein, what we call polypeptides simply, of different nature. For example, 16S rRNA in case of 30S of prokaryotes, whereas 18S RNA in case of 40S of eukaryotes. The same 23S and 5S in case of prokaryotes in larger subunit, and in case of larger subunit, that means 6S of eukaryotes contain 28S RNA, 5.S RNA, 5S RNA plus nearly 49 polypeptides. So that means it's a clear-cut example that they form the structural components of ribosomes with respect to ribosome RNA. And how they get synthesized, this is very important. It's about basics, we all knew that. How they get synthesized, synthesis is very important. Say in case of eukaryotes, 28S, 5.8S and 18S rRNAs. They are synthesized by RNA polymerase 1. They are synthesized by RNA polymerase 1 and their transcription units, that means the genes concerned with 28S, 5.8S and 18S rRNA are present in nucleolus in the way of multiple copies. Say simply gene clusters. Multiple copies. Gene clusters are present, that we simply call. Gene clusters are present. Whether they form directly, no. The first formed rRNA is called pre-rRNA. But that means gene clusters they form single pre-rRNA. What we call 45S rRNA. That means a combination of these present in 45S RNA. It has been cleaved by ribonuclease activity to form 32S and 12S RNA that again cleave to form 28S, 5.8S and 18S rRNA. That means these are called precursor RNAs. rRNAs. And this one is called pre-rRNAs. That means from pre-RNA, that is from gene cluster it form as a single pre-RNA, then the precursor RNA, then the final matured RNA. This is how it's about the gene genetics. How they get synthesized, whether in one copy, no, in multiple phases. Then with respect to 5S rRNA, in case of eukaryotes, it will be synthesized by RNA polymerase 3. Same, multiple copies is also formed. That means multiple copies of genes are present. And the most important component is, in case of eukaryotes, like trypanosoma, there is a pre-RNA which contains introns. You know, introns has to get removed, what we call excision. How it is done? It is done by self-splicing. Self-splicing is reported in case of eukaryotes. Self-splicing. That means ribosome activity of RNA. How it is performed? By rich G residues. The rich guanosine residues present in the pre-RNA itself cut down the introns, what we call self-splicing, is reported in eukaryote like trypanosoma. This is very important point. Self-splicing means RNA itself have, that means ribosomal activity. You know, during evolution process, during those days or pre-life days, you know, RNA has the catalytic activity. It's called ribosome. Later onwards, during evolution, its catalytic activity has been shifted to the enzymes. That means the RNA are simply the RNA component in the ribonucleoproteins which have the catalytic activity is called ribosome. And still it is present in certain eukaryotes, you know, which are through self-splicing. This is very, very important component, you know. This is how the issue ends. This is with respect to RNA. And the final one, that is transfer RNA. Transfer RNA, what we call. Hole proposed a cloverleaf structure. On average, it contains 75 nucleotides. This is T arm. This is D arm. This is acceptor arm, what we call amino acid arm. This is anticodon arm. For example, this is mRNA, what we call codon anticodon interaction. In genetic code, when we discuss about this, we have to discuss about one important point, genetic code degeneracy. Then we can extend about this concept. Okay, for a while, just think, just listen. Anticodon interaction means, say AUG, for example. Then there's UAC, is complementary base pairing. That's how the issue ends. You know, codon anticodon interaction. Say that this is simply called clover leaf model. The secondary structure. It is a secondary structure. It is an adapter molecule. You know, tRNAs. When we discuss about genetic code, we can speak at length. Actually, tRNA transfer, what it transfers? It transfers a specific amino acid to the ribosomal machinery during the protein synthesis. That is why they are considered as adapter molecules. There are different M's and the acceptor arms, T arm, anticodon arm, D arm, and variable arm. Based on variable arm, they can have two classes. Class 1 tRNAs and class 2. Okay, it's not too much important, but D arm, for example, what is D? Dihydrouridine. There are some unusual bases are present. Say 1-methyl guanosine. pseudouridine, pseudouridine, for example, pseudouridine, 1-methylguanosine. Because based on that, the most important point is the anticodon, which contains some 7 unpaired single nucleotides, out of which 3, 4, 5 act as anticodon, which are in complementary to the codon of mRNA. You know, codon means simply a triplet nucleotide sequence which have a sense. That means a codon for one amino acid. We'll discuss in genetic code, okay? So it's about tRNA. This acceptor arm, this is very important, at 3' end, what we call CCA end. It will bring, just like, you know, this is one amino acid, my head is acceptor, the CCA end. It will hold one amino acid and will bring to the missionary, ribosomal missionary, that's it. And each tRNA is specific to one amino acid. That means, okay, for example, there is one tRNA which can recognize more than one single codon. That means, though there are 61 codons, but the number of tRNAs are just 40. What exactly it means? Okay, through genetic code in a correlation man, we'll try to understand. Okay, this is about the tRNA. Simply, it will transfer or adapter molecule which will get the specific amino acid to the protein machinery. That's it. Okay, and CCN it will carry, whereas anticodon which participate in codon anticodon interaction during protein synthesis. This is very important point. Okay, there are certain unusual bases like pseudouridine and 1-methylguanosine. Okay, it's about tRNAs. Then the most important part is genetic code. What is genetic code? For example, you know, this is mRNA. So this is AUG. This is UUU. This is A. This is GGG. And this is CCC, etc. For example, AUG codes for methionine. UUU codes for phenylalanine. A codes for lysine. CCC codes for proline. GGG codes for glycine. That means a triplet nucleotide sequence. Simply useful in DNA language to a protein language. That's how the main aim of genetic, what is genetic code? It's a triplet nucleotides which aids in the conversion of a DNA information to protein, that means polypeptides, through a codon which have a specific language. Each codon codes for one amino acid. Now, there are, for example, you know how it works. There are 4 nucleotides. The possible number of codons out of 4 nucleotides is 61, what we call sense codons. And most important point, more than one codons, or simply more than one codon codes for one amino acid. How it works? Say we have 20 amino acids, what we call standard amino acids, and we have 61 codons. That means one amino acid is coded by more than one. For example, glycine has 4 codons. Glycine has 4 codons. Here the most important point is, though more than one codon codes for one amino acid, but it does not mean one codon codes for more than one amino acid. There is a specificity, what we call. In the way of properties we can discuss. This is very important point. in the what we call specificity. Specificity. That means one codon codes for one particular amino acid. That's it. And genetic codes is completely unambiguous. Unambiguous. Because it is comma-less, punctuation-less. For example, how it works. You know, simply it starts. We have to start from one end, fixed end. Say U, G, C, C. How we have to read? Say U as 1, G as 1, C as 1, A as 1. It is phenylalanine. This is glycine. This is proline. This is lysine. That means it is continuous. There are no punctuations and or commas. This is simply unambiguous. And most important point we have to discuss whenever the genetic code comes, what we call verbal hypothesis. Verbal hypothesis. Verbal hypothesis. What exactly the verbal hypothesis? We have already discussed more than one codon codes for one amino acid. How it works? You know, for example, there is one codon. The corresponding complementary is, in case of tRNA, anticodon is AAA. The thing is, the first two bases present in mRNA codon participate in the complementary base pairing with that of the last two bases of anticodon. That means the third base has been left out. But because of loose or lenient or flexible base pairing. That means 3' base of codon is not, we cannot recognize the 5' base, which is the 5' base of anticodon present in tRNA. It's a hypothesis, what we call verbal hypothesis. Verbal, the third base is verbal. Crick proposed this, and they attributed it to the spatial differentiation of the 5' end in tRNA. Spatial differentiation, you know, spatial differentiation of 5' in tRNA, 5' ends. Because of this, the third base present in the codon cannot recognize the complementary base pairing in anticodon of tRNA. That is why what is the verbal position is a third. What we call lenient or flexible position in codon, the third position. The third position is called verbal position. In a codon, for example, this codon, the third position is called verbal position. And how it ends? tRNA, that means a single tRNA can recognize more than one amino acid. How this end? There are nearly 40 tRNAs for 61 sense codons. You should carefully understand. There is a specificity of genetic code. One codon recognizes one particular amino acid, but it does not mean that for one amino acid there are only one codon. For glycine there are 4 codons. Though there are 4 codons for glycine, but each are, that means all the 4 codons are specific to glycine only, and they won't recognize another amino acid. This in case of tRNA, the same. The possible number of tRNAs are 40. That means it can recognize more than one codon. Simply what we call. So that means it can recognize more than, so simply the correlation is same. 40 tRNAs, 61 codons. That means one tRNA can recognize more than one codon. That means the collinearity is same. One tRNA, that means tRNA is specific to one amino acid. tRNA is specific to amino acid, but tRNA can recognize more than one codon. This is very important. It's a master stroke. One tRNA are specific to amino acids, but tRNA can recognize more than one codon. Here, more than one codon can recognize one amino acid. This is very, very important. And generally, in exams, they will ask a question on this because it is somewhat twisted. Okay, it's about verbal hypothesis, a quick propose this, and it is a major important aspect. And it is because it explains about degeneracy of codons. Degeneracy of codon. That means more than one codon can code for one amino acid. And how it was? Because of verbal hypothesis. Because the third base in a codon is flexible or linear because of spatial arrangement of 5' in case of 5' base in case of tRNA. It's what we call attribution, what we call verbal hypothesis. So the third base in a codon is verbal position. And one more important property of genetic code is genetic code is almost universal. Genetic code is almost universal, nearly universal, because it's a terrific feature. Because over the evolution, throughout the ages, the genetic code has been conserved except certain exceptions. For example, a mitochondria, in mitochondrial mRNA, we know there is a codon UGA. It codes for tryptophan. Actually, UGA codes for stop in case of nuclear. In case of mitochondria, UGA codes for tryptophan, not a stop codon. That means genetic code is not absolutely universal. It is universal but having some exception, what we call almost universal. This is very important. Another example, take AGA, which actually codes for arginine. It actually codes for arginine, but in case of mitochondrial mRNA, it's a stop codon. Arginine in case of nuclear mRNA, AGA is a genetic code which codes for arginine in case of nuclear, but in case of mitochondria, it's a stop codon. That means genetic code is nearly universal. That means genetic code degeneracy. That means more than one codon for one amino acid, and one tRNA can recognize more than one codon, and one tRNA for one amino acid because of verbal position, the third position, because there is a lenient or flexible nature of third position because of spatial differentiation. And genetic code is comma, that means is unambiguous. Specificity, one codon for one amino acid. And genetic code is an essential component which translates the DNA language to a protein language. This is how this is about different issues. What I would like to suggest is, though my classes serve you in terms of basic and advanced issues, you have to follow one standard reference and one basic book. For like Fulfilled or Leninger for extension, and my petty contribution aids you in further elaboration of the topic. So my lectures plus one reference book, one standard book may help you in clearing top most exam like NEET, CSR. I already told my objective is to serve for the poor and needy students. You may like and comment, and thanks for watching.